Bismillahirrahmanirrahim. This is the third video on the March 2022 paper, and this is going to be, we're dealing with question 5 and 6 in this. In this video, we start with question number 5. Factor VIII can be made as a recombinant human protein. Name the disease that is treated using recombinant human factor VIII. Of course, you know that it is hemophilia. You should know that. I hope you know that. Before recombinant human factor VIII was available, this disease was treated with factor VIII from donated blood. Give two advantages of using recombinant human factor instead of factor VIII from donated blood to treat this disease. Naturally, large unlimited supply available, no risk of infection, there are few ethical issues, then less chance of rejection, then it's cheap and quick and easy to produce, and no need to wait for donors. So you could have come up with a lot of stuff, because we would be depending on donated blood. Now, in this case, we will not be depending on donated blood, so we don't need that. So easy to write, large unlimited supplies, no risk of infection, no ethical issues, no immune response, or no allergic reactions or side effects, and then cheap and quick to produce, and no need to wait for donors. You see, the risk of infection, this point has to be clear. You see, we don't know what we can transfer. We can transfer many viruses in donated blood, and that is how people used to get HIV when we used to give them blood transfusions from HIV infected people's blood. Okay, then we come to the B part of the question. The gene that codes for human factor VIII can be synthesized from messenger RNA purified from human liver cells. Name the enzyme that uses messenger RNA as a template to produce complementary DNA. Now again, this is very much part of the new syllabus, and that's reverse transcriptase. Well, of course, it was in the old syllabus as well. Reverse transcriptase. Right? Now, part 2 outlined two sources other than messenger RNA from which genes can be obtained for genetic engineering. So either you could have cut it from DNA, because we know the genes which are present on which chromosomes, you can just cut them by using restriction enzymes, or you can synthesize genes from nucleotides, because if you know the amino acid sequence, then you know the DNA sequence and you can synthesize it from nucleotides. Then coming to part C of the question, the gene that codes for human factor VIII can be transferred into mammalian cells in tissue culture. Explain why a promoter also needs to be transferred into the mammalian cell so that human factor VIII can be synthesized. You see, in order to have gene expression or in order to start transcription, you have to have a promoter, because that is the area which is recognized by the RNA polymerase. So transcription factor can bind to the promoter. So then RNA polymerase can bind to the promoter, and then you produce the correct quantity or promoter for the marker gene to show the gene transfer is successful. So the reason why you put promoters, and this is for 2 marks. So you must know the effect of promoters and why do we need to have promoters when we are going to use recombinant DNA technology. Required for gene expression, so transcription factor can bind to promoter, so RNA polymerase can bind to promoter, produce the correct quantity / inducible / promoter for marker gene to show gene transfer is successful. Sometimes we have to put a marker gene next to it so that we can then identify which ones have been, where has it been taken up. Now coming to question number 6. A part, the banana plant, whatever, Musa cuminata, is a tall herbaceous plant with very large leaves. Figure 6.1 shows a banana plant. I'm sure you've seen one. We have a lot of them growing here in Lahore, Pakistan. An investigation was carried out to measure the net carbon dioxide uptake by a banana plant at different light intensities. So immediately you know this is a question on the chapter on photosynthesis. Figure 6.2 shows the results of the investigation. As I always say, look at the graph very carefully, but then come back to the graph when you have read the further question of it. So always read the whole question. Look at the graph now. I'm not saying don't look at it now. Look at it now, but then read the question and come back to it again. So now what do we have here? We have mean net carbon dioxide uptake in micromoles per meter square per second. Look at the units. Micromole per meter square per second. And here on the x-axis, what do we have? We have light intensity and the unit is lux. But here in the mean net carbon dioxide uptake, you see you've got -0, -2, -4, and then you have +2, and then +4 and +6 like this. So look at it very carefully, and then we have got, of course, got +8 as well. It isn't saying, but that's what it implies. Then let's look at the question. Now the question is very, very basic. It's come many times in your syllabus. I've seen this question many times. With reference to figure 6.2, so we go back to figure 6.2, describe and explain the results at a light intensity of 1000 lux. Then part 2, with reference to figure 6.2, describe and explain what can be concluded from the graph at light intensity between 2000 and 7000 lux. And then we read the further, part 3, explain why the rate of carbon dioxide uptake levels off as light intensity increases above 10,000. So we're going to be talking about 10,000, 7,000, 2,000, and 1,000. So let's go back to the graph and let's look at it, and let's look at it now very carefully. So we're going to be talking about number 1, 1,000 here, then 2,000, then I think we have 7,000, so that is going to come here, and then we're going to talk about 10,000. So look at all these, draw lines on the graph. I would like you to draw a line on the graph here, then on 2000 here, and then on 7000 here, which goes right up, maybe not very straight, and then 10,000 here. So you go straight up and you see where it hits the graph. Now, if you've done this, you've understood it, the next thing you've got to do is you've got to give me figures. So you need to read off this point, and this point, wherever it is. I'm not, maybe not very correctly drawing the horizontal line. So first is, with reference to figure 6.2, describe and explain the results at light intensity of 1000. And this is 3 marks. So be very careful what you write here, because if you write something here and then you write the same thing here, well, then there's something wrong. There cannot be 1 mark scheme for the same stuff you wrote here and the same stuff you wrote here. So please remember, you have to be careful what you write. What you write here should not be duplicated in the second part, because in one place then it is wrong. So at 1000 lux, what do we have here? 1000 lux here, you see this point here. I'm just going to draw it again. 1000 is here and then here. So 1000 is at light intensity increases, rate of CO2 uptake. What is happening? CO2 uptake below is 0. Is here in this situation at 1000, CO2 uptake is below 0. Because here we have -0 and then we have 2 up there. So it is -2 or something. It's not even -2, it's minus 1.6 micromole per meter square per second. It is minus 1.6 micromole meter square per second. So what does this mean? This, you've given me the figures. Little photosynthesis is occurring. You can't say no photosynthesis is occurring. Little photosynthesis is occurring, and due to low light intensity, but the rate of respiration is higher than the rate of photosynthesis. So mean net carbon dioxide is a negative. So this is what you had to give me for the 1000. So carbon dioxide uptake is below 0. That is, carbon dioxide uptake is minus 1.6 micromole per meter square per second. Then little photosynthesis is occurring. And please remember here, now I've written this in red, reject no photosynthesis, because there's still low light intensity. So is reject no photosynthesis. We didn't say it's totally dark. Reject no photosynthesis. That means you can't write that. And the reason why there is this less photosynthesis is because there's low light intensity, this point here in the mark scheme. And the reason is the rate of respiration is higher than the rate of photosynthesis. So there's little photosynthesis occurring, but respiration, of course, will take at a constant rate. Respiration occurs during the day and at night. But at this point in time, with little light, like it happens at dusk and dawn, the rate of respiration is higher than the rate of photosynthesis. So more carbon dioxide is being produced. There is no carbon dioxide uptake as yet. It's on the negative because it is on the y-axis. What did we have on the y-axis? It mean net carbon dioxide uptake. So it's negative uptake means it's being produced. Now we come to part 2 of the question, which we've read before as well, with reference to figure 6.2. Describe and explain what can be concluded from the graph. What can be concluded from the graph at light intensity between 2000 and, so this is the 2000 mark and this is the 7000 mark. So in this you can see what is happening. The rate of photosynthesis is increasing and it's increasing. It's constantly increasing. So that means what is on the x-axis? Light intensity is the limiting factor. So as light intensity increases, rate of CO2 is increasing. Data quote you give me a data quote 2000 lux is 0.2 minus 0.2 and 7000 is 6.5 micromole per meter square per second. You have to give me the units every time. Please remember that. Now the reason is the rate of photosynthesis is increasing, but the main reason is that the light dependent reaction is increasing. Also the light independent is also increasing because photosynthesis is dependent on these two reactions, light dependent and light independent. And here, of course, as I said, what is on the x-axis? Light intensity. If you say light, I'm sorry, I'm not going to give you a mark. Light intensity is the limiting factor. So how would you word it? As light intensity increases, rate of CO2 uptake increases, then the data, 2000 lux minus 0.2, 7000, 6.5. Remember units. You must always remember the units because light dependent and light independent reaction both are increasing. Light intensity is limiting factor. So light intensity more means more ATP, more NADPH means what? More of the Calvin cycle taking place. So both light dependent and light independent reactions are going to increase. Then coming to the part 3 of the question, explain why the rate of carbon dioxide uptake levels off as light intensity increases above 10,000. Yeah, naturally light intensity is no longer limiting. Now some other is limiting, like CO2 concentration or temperature is now the limiting factor. So light intensity no longer limiting because now it has become constant, and now some other factor is limiting, CO2 concentration or temperature. Then we come to the B part of the question. Pitcher plants are carnivorous plants that trap and digest insects in a large modified leaf called a pitcher. Figure 6.3 shows a pale pitcher plant, whatever. Pitcher. Figure 6.3, let's look at it carefully. When we look at the diagram, there it is. Unlike the Venus flytrap, pitcher plants have no moving parts to trap insects. Insects are attracted to scent produced at the top of the pitcher. Once inside the pitcher, the insect slides down to the bottom and into a liquid containing digestive enzymes. So, I mean, it's really making a fool of the insects because they are attracted to the scent and they move into the pitcher and the insect slides down, must have a very must have a very sticky or slippery slope of the sides, and then it falls to the bottom and into a liquid containing digestive enzymes. Suggest a type of enzyme found in the pitcher plant that is used to digest. Now, of course, what are these enzymes going to be? They're going to be hydrolytic enzymes because we want the insect to be digested. So hydrolytic, you could say extracellular, you could have said a protease, you could have said proteinase, you could have said a peptidase. So you could have said all these because basically what is an insect? It's like a meat. It's a protein. Like when you're eating chicken or when you're eating fish, you need proteases. Then, of course, it comes to part 2. Pitcher plants grow in bogs and wet grasslands where the soil has a low concentration of minerals such as nitrates. The plants obtain nitrates from the digestion of insects. Calvin cycle intermediates are used to synthesize more complex compounds, some of which need nitrogen from nitrates. Name one compound that needs nitrogen from nitrates and name the Calvin cycle intermediate from which it is synthesized. Compound that needs nitrogen, well, of course, we all know that. Amino acids, proteins. Amino acids, or you could have said proteins. And of course, one very important one, nucleotides. Nitrogenous base. And then the Calvin cycle intermediate is GP. Now, why is it GP? It's actually a triose phosphate. So you could have said TP as well. GP or TP, glycerate 3-phosphate or triose phosphate. because the 3 carbon that leaves the Calvin cycle is a 3 carbon, that's a triose phosphate. And then we tell you that if the Calvin cycle goes twice, then you get a glucose, C6H12O6. So it's a 3 carbon C3H6O3, C3H6O3, yes. So triose phosphate would be the one which is the Calvin cycle intermediate, which will ultimately be formed. Two of these will form a glucose molecule. So that was the Calvin cycle intermediate which you have to give me. Question 7. In aerobic respiration, ATP is synthesized by substrate-linked reactions and by oxidative phosphorylation. You know substrate-linked reactions take place in the glycolysis and in the Krebs cycle, while oxidative phosphorylation takes place in the ETC or the electron transport chain. So outline the process of oxidative. Now that's a very direct question, and you used to get that in the essay questions, but this is the direct question, and this is for 7 marks. So you've got to understand is you've got to give me enough points to get the 7 marks which are in this part of the question. And I think everybody can get this. This is a very easy question. Now, as you can see, there are 11 points, and out of these 11, you have to give me any 7. So I'm sure you kind of remembered this. It's all something which you have to remember. So reduced NAD and FAD, where have they come from? They've come from glycolysis, they've come from the link reaction, they've come from the Krebs cycle. Now, as they come here to the ETC, they release their hydrogen, and the hydrogen splits into a proton and electron. Now, where is this happening? On the inner mitochondrial membrane or the cristae? Then the electrons pass through the electron transport chain. They pass from one carrier to the next, and a little bit of energy is released in this process, which of course helps in pumping the protons into the intermembrane space. So this is the point here. A proton gradient is established. What does that mean? That more in the intermembrane space and none in the matrix. Now the protons, they have only one way to come out, and they can only diffuse through the ATP synthase. And here, when the protons move through ATP synthase, an enzyme is present at the base of ATP synthase. You can see it's A, so it's an enzyme. And here, ADP and inorganic phosphate combine to form ATP. So the protons diffuse through the ATP synthase, and ATP is produced from ADP and inorganic phosphate. This is called chemiosmosis, and oxygen acts as a final electron acceptor, and ultimately water is formed. So out of 11 of these points, you have to give me any 7 and you would get your 7 out of 7. Now coming to the B part of the question. In an experiment on respiration, 2 different populations of yeast cells were used, A and B. Yeast cells in population A had no mitochondria in their cells. Yeast cells in population B did have mitochondria in the cells. So A, no mitochondria, B did have mitochondria. Both populations were provided with glucose in solution, and the concentration of ATP was measured every minute for 7 minutes. Concentration of ATP was measured every minute for 7 minutes. So ATP concentration is on the y-axis, and on the x-axis, what do we have? Time. Because it was measured every minute, 1 minute, 2 minutes, 3, 4, 5, 7, for 7 minutes. And this is the time in minutes. Do this story with yourself when you're reading the paper 4. Paper 4 is a very difficult paper, and you must develop the right skills to do this paper. There's nothing you can learn from the books. It is a skill of exam skill which you must have. So now you look at population A and population B. Look at the graph, and what have I told you? You read the question and then you come back to the graph. So population A and population B, you can see ATP concentration is much higher in population B. What was population B? They had mitochondria. Population A did not have any mitochondria. So you can see how it is increasing. The ATP concentration is gradually increasing in A. And then you can see it's gradually increasing in here. Then there's little increase more, and then it's sort of remaining the same. While in B, which had mitochondria, you can see there's a lot of increase, then a lot of increase, and then increase again. Now let's look at the questions. Describe and explain the difference in results between population A and population B as shown in 7.1. Describe and explain. Describe is no biology, explain is a lot of biology. So describe and explain the difference, and this is for 4 marks. This is the easiest 4 marks that you could get in any paper 4. Remember this, these are the easiest paper 4 marks, and you can easily get these. Now when you look at these, let's look at them now carefully. Now the differences are what? Differences are population B produces more ATP. That's very obvious. Population B produces more ATP than population A. Or you could have said the reverse, A produces less ATP. Then production of ATP increases at a higher rate for population B. Yes, you can see that as a rate is a higher rate. Then you give me a data code, paired data code. Then population B carries out glycolysis and substrate link phosphorylation and oxidative phosphorylation. Why? Because B had mitochondria in them, so they have mitochondria in them. And population A only does what? There's an explanation. Population A carries out only glycolysis because respiration in anaerobic conditions. So there's only ethanol fermentation or substrate link phosphorylation. Or you could have said they cannot carry out oxidative phosphorylation because they have no mitochondria. So the wordings would be population B produces more ATP, production of ATP increases at a higher rate for population B. Then population B carries out glycolysis and oxidative phosphorylation because it had mitochondria. So it will carry out both the processes, glycolysis and oxidative phosphorylation. While population A is pathetic, it has no mitochondria, so it can only carry out glycolysis because cytoplasm is present. So glycolysis takes place in the cytoplasm. Or you could have said cannot carry out oxidative phosphorylation. Now the data code looks something like this. But of course, you're supposed to give me one. I'm not saying you have to give me all this. So, but then this helps you to understand how you have to give me the data code. So time at 0, time at 1 minute was A was 2 and B was 10. Then you have to give me the units as well. And then of course, if we calculated the rates, then these were the rates which we calculated at the end of it all. So I've given you all the data code and you need to check it with a graph. That's why I've not drawn anything on the graph. And check it with the graph and see how we've got this data code and how we have calculated. This will be the overall rate which we have calculated. And how have we calculated this? Please, everybody needs to figure this out. Now, coming to the eighth question, natural selection and artificial selection, selective breeding, both have important roles in evolutionary change. Seahorses are fish with an unusual appearance and many specialized features. I've always been very, very enamored by seahorses. I think they're so beautiful. Seahorses are fish with an unusual appearance and many specialized features. Two species of the seahorse, Hippocampus erectus and H. zosteria, are found in the coastal waters of the Gulf of Mexico and the Caribbean Sea. So there are two species of seahorses. It is thought that these two species of seahorses have a common ancestor. The range of the two species overlap in many areas. Male and female seahorses stay together for the whole of the breeding season. H. erectus is much larger than H. zosteria. Few seahorses occur that are intermediate in size between the two species. So we have the larger ones, and so much larger than the other. So we've got a larger one and a smaller one, and then few seahorses are intermediate in size between the two species. Use this information to name and explain the type of speciation that may have occurred in the evolution of these two species. So basically, what have we to understand? Explain the type of speciation. The type of speciation, they're in the same place. They haven't been divided by geographical barriers or by land or by water or something. So this would be sympatric speciation, because the other one is allopatric, which is, I say, when they're apart and there is some geographical isolation. There's no such thing. So the name and explain. So the name would be sympatric speciation, and that would get you your 1 mark for this. And now the explanation, you will have to start with the explanation. There was there is no geographical barrier, so that is why it was not allopatric speciation. And this is more of a reproductive isolation and behavioral isolation. So these are now points of the new syllabus, which they are now expecting you to be able to talk about. These are not the old syllabus. So sympatric speciation is behavioral more and reproductive isolation and behavioral isolation. Part B, for many years, farmers have used selective breeding to improve the milk yield of dairy cattle and to improve the yield of crops. Explain why improving milk yields in cattle by selective breeding can be more challenging than improving yields of crops by selective breeding. What should come to your mind is that in one situation we are handling animals, improving yield in cattle, and the other we are just doing crops. So it's very difficult to compare the two. They're two totally different things. It's a longer time to grow. I mean, animals take a long time to grow. Crops are only 3 to 4 months crop, better wheat crop, better sugarcane crop. Then fewer offsprings per cross. Cattle is going to breed only give you one or two calves. Fewer offsprings per cross. Feature can only be measured in about 50% of offspring, which are females, because we're only measuring milk is only given by the females. The male doesn't give us any milk. Milk yield must be measured over a period of time. Then there are ethical issues. Then difficulty in identifying suitable males for use in these crosses. Then cattle require more care and more support, and there's more expensive. We need to keep them for longer periods of time before they even start producing milk. And it is more dangerous to carry out breeding of large cattle. So these were the points that you had to give me. So the wordings of the question or the answer would be takes longer time to grow. Cattle will take a lot of time to grow, then there are fewer offsprings per cross, then features can only be measured in females, about 50% of the offsprings. So milk yield must be measured over a period of time. Then there are ethical issues. You can't be doing things to animals just like that and doing something which is very wrong ethically. Difficulty to identify males for use in crosses because you have to look at their female relatives. And then more expensive to care for cattle. Crops are not that expensive, you just have to put fertilizer and water them. Then coming to the last part of the question, state two examples of crop features that may be improved by selective breeding to increase the yield of crops. Two examples of crop features that may be improved by selective breeding to increase the yield of crops. So the points could have been the mass or the quantity of the crop or the fruit that we want to harvest, and the disease resistance of the crops, then resistance to insects or pests, then rate of growth should be very, very fast. Then drought resistant, they should be able to survive long periods of drought without rain or water. Then resistance to herbicides, and then resistance to flooding and wind. They should have strong stems so that they don't topple over in the wind, or they can resist a lot of, if there is flood and there's standing water for some time, then they can resist that. I complete this video here, and there are two more questions left, and I'll continue that in the next video. Thank you very much. Thank you for watching. Please do tell your friends and please do subscribe. Thank you very much. We have completed question 5, 6, 7, and 8 in this video.